Hey everyone! In this analysis, we will be examining a 2025 paper by Liu and colleagues titled Human Umbilical Cord Plasma Metabolomics Uncover Potential Metabolites for Combating Aging, which was published in the journal Aging Cell. Here is our agenda. We will begin by contextualizing the research into UCB's potential. Then, we will detail the study's metabolomic findings and the process for candidate identification. After that, we'll review the experimental validation in both cellular and C. elegans models. And we'll conclude with the study's overall significance and future directions. All right, let's start with the first section, HUCB's rejuvenating potential. First, we need to establish the scientific background that motivates this type of investigation into umbilical cord blood. The foundational research in this area utilized parabiosis models. This is a technique where two organisms are surgically joined to share a single circulatory system. These studies demonstrated that circulating factors in young blood can reverse age-associated functional decline across multiple organ systems in aged mice. Building on this, subsequent studies have shown that human umbilical cord blood, or HUCB, which is the youngest form of human plasma, can enhance cognitive function in aged mice. So, while the rejuvenating potential of HUCB is established, the specific small molecule metabolites responsible for these anti-aging effects have, until now, remained largely uncharacterized. And this leads us to the central question that forms the primary objective of the Liu et al. Study. Which specific metabolites within human umbilical cord plasma are the ones that confer its anti-aging properties? Okay, let's move on to the study's core methodology and its initial large-scale findings. This section is on the cord plasma metabolome, with a comparative analysis of neonatal and adult plasma. The study employed a robust, multi-cohort design. They collected plasma from 330 participants, divided into 4 groups: neonatal cord blood, a young adult group, a middle-aged group, and an elderly group. They then conducted untargeted metabolomic profiling using UPLC, ESI, MS/MS to identify and compare metabolites across these different age groups. This allowed for a comprehensive comparison across the human lifespan. The analysis revealed a distinct metabolic landscape in cord plasma. In total, 1,092 compounds were detected. Of these, 662, that's over 60%, showed a significant difference between the groups. Furthermore, looking at metabolic pathways, 43 out of 59 analyzed pathways were altered. These are substantial differences at both the compound and the pathway levels when comparing cord plasma to adult plasma. An important finding was that the top 10 most significantly different metabolic pathways all exhibited higher metabolite abundance in cord plasma. This includes pathways such as beta-alanine metabolism, histidine metabolism, and thymine metabolism. What this indicates is a systemic elevation of specific metabolic activities in neonates compared to adults. So now we'll examine how the researchers narrowed down this extensive dataset in order to identify metabolites with the highest anti-aging potential. This involved a 2-step filtration approach. A 2-tiered bioinformatic filtering process was applied. The goal was to isolate metabolites that are both highly abundant at birth and also decline progressively with age. The first step was a correlation analysis to identify metabolites whose abundance was negatively correlated to age. The second step then filtered for metabolites that showed a significant decrease in adult blood compared to cord blood. This systematic approach yielded a final list of 211 candidate metabolites, which were then prioritized for further investigation. Functional annotation of these 211 candidates was then performed. It revealed that these metabolites are associated with 5 key biological processes: inflammation, oxidative stress, energy and nutrition metabolism, proteostasis, and the DNA damage response. These are, of course, all considered to be hallmarks of aging. The investigation then proceeded to test the biological activity of the top candidates. For this, they used established cellular models of senescence to validate any anti-senescence activity in vitro. A formula, which they designated Cord Formula 1, or CF1, was created from 5 of the top-ranked metabolites: carnosine, torocalic acid, inosine, L-histidine, and N-acetylneuramic acid. The efficacy of CF1 was then assessed in a human embryonic lung fibroblast cell model, MRC5. Senescence was induced using 2 different stressors: etoposide to cause DNA damage and hydrogen peroxide for oxidation stress. The results showed that treatment with CF1 significantly attenuated senescence. This was evidenced by a reduction in SA beta-gal positive cells, a key senescence marker, and also by the suppression of key senescence-associated genes, including P16 and P21. All right, to assess the effects at an organismal level, the anti-aging potential of CF1 was next evaluated in the model organism Caenorhabditis elegans. The results from this model demonstrated that CF1 treatment significantly extended both the lifespan and the health span of the nematodes. Health span was measured by key physiological functions that normally decline with age, such as body bending frequency and pharyngeal pumping rate, both of which were significantly increased in the treated group. Further analysis in the C. elegans model confirmed additional benefits. These included a reduced accumulation of lipofuscin, which is an age-related pigment, as well as enhanced resistance to heat shock stress. Importantly, there were no observed adverse effects on fecundity or body size, suggesting the formula does not compromise reproductive fitness. Finally, let's consider the broader significance of this research and the future work it enables. This brings us to the implications of the study's findings. To summarize, this study makes several novel contributions. First, it provides the first comprehensive comparative metabolome of human umbilical cord versus adult plasma. Second, it identifies a specific set of 211 cord-enriched, age-declining metabolites with anti-aging potential. And third, it delivers proof of concept that a formulation of these metabolites can extend both lifespan and health span in model organisms. The authors themselves highlight the high translational potential of these findings. They state that endogenous metabolites offer inherent advantages for anti-aging intervention development, including favorable bioavailability and safety profiles. Their natural presence in human neonates may streamline future preclinical and clinical development. This points to the fact that because these are naturally occurring human compounds, the path to therapeutic development could be more direct. So the study concludes by positioning these findings as a foundation for future research. It leaves us with a key question for the field. Are endogenous metabolites, particularly those that are abundant in early life, a viable therapeutic avenue for extending human health span? The data presented here certainly suggests this is a promising area for continued investigation.